Drugs have been around since the beginning of time, and to no surprise, they are still used and sold to this day. Cocaine is one of the many addictive drugs that can lead to life-altering events. These life changes could include the loss of a career, becoming financially unstable, and even becoming homeless. Cocaine's ability to alter the way a person thinks and moves is one of the many terrifying side effects that this brings. It is without a doubt that this is a serious issue for people all over the world, and initiatives are being taken to find a solution to this drug problem. Some of these ideas to eliminate effects caused by cocaine include targeting specific dopamine receptors that can otherwise create the addictive feeling caused by cocaine. The way cocaine enters the brain is through the bloodstream. Once it has reached the brain, it causes a buildup of the neurochemical dopamine. Dopamine acts as a regulator for many nerve cells in the brain, and they are responsible for maintaining the right level of activity to achieve our needs and goals. When a certain task requires our muscle or mind to work harder, dopamine initiates some of the brain cells to take on these challenges. Dopamine comes from a set of brain cells called the dopaminergic cells, which make dopamine and sends the chemical into surrounding areas. These free-flowing dopamine molecules are then attached onto receptor proteins, which can then stimulate the cell receptors and alter electrical impulses, therefore altering the cell functions. To keep the altering of cell functions at a balance, the dopaminergic cells pull some of the previous dopamine molecule release back into themselves. However, cocaine interferes with this mechanism. Cocaine inhibits the dopamine transporter, a protein that the dopaminergic cells use to bring back the dopamine molecules. As a result of this, dopamine is not used and causes a buildup. The way cocaine creates its addictive effect and changes the mental state of an individual is primarily through its effect on the limbic system. The limbic system is the region of the brain where pleasure and motivation are regulated. A part of the limbic system, called the nucleus accumbens, produces feelings of pleasure when stimulated by dopamine. Since cocaine can tie up the dopamine transporter, causing a buildup of dopamine, this will lead to a boost of dopamine to the nucleus accumbens, thus causing a rushing feeling of pleasure. These feelings of pleasure can become addictive and create a sense of wanting for more cocaine. In this experiment, researchers tested to see whether drug-related environmental stimuli could play a factor in causing a relapse on cocaine after a long duration of abstinence. Along with this, they also tested to see whether a D1 receptor antagonist, also known as SCH39166, affected the results. To start off, rats were trained to associate two discriminative stimuli, IV cocaine versus non-rewarding saline solution. The rats self-administered IV cocaine while at the same time learned the difference associated with cocaine availability and unavailability. Once cocaine level intake became stable, the rats were given either cocaine or saline as the only available stimuli during each session. These stimuli were then taken away and withheld. Later, re-exposure to the discriminative stimuli were measured in two different groups of rats to see if any changes had occurred. In the first group, the rats were tested immediately after stimuli were taken away, while in the second group, the rats were confined in their homes for additional 4 months before testing. After testing from both groups, rats were then divided into three groups where a test was conducted with the cocaine stimuli, saline stimuli, and cocaine stimuli, followed by the administration of SCH39166. In the test for the removal of stimuli, both groups showed that only the cocaine stimuli had a strong relapse. In the re-exposure test, the addictive effects from cocaine was reversed by selective dopamine D1 receptor antagonists in both groups of rats. Results from this study have shown that addiction from cocaine did not diminish and relapse risk is still at a high even after a certain period of time. However, rats induced with SCH39166 showed a significant improvement in cocaine-seeking behavior of the rats. This means that further research on dopamine receptor antagonist drugs could lead to a solution for cocaine addiction. In the second study, researchers hypothesized that the dopamine D3 receptor played a role in cocaine addiction. The dopamine D3 receptor antagonist was induced to laboratory rats, and the measurement of cocaine-enhanced electrical brain stimulation reward, cocaine-induced condition-placed preference, and cocaine-triggered reinstatement of cocaine-seeking behavior were taken from the rats. The drugs in the study, SB277011A, has significant affinity in both human and rat D3 receptors. In other words, this drug is highly attracted to the D3 receptor. In this experiment, male rats were housed individually from the cocaine trigger reinstatement and electrical brain stimulation reward experiment, while rats from another species were used for the cocaine-induced condition-placed preference experiment. This was due to another institution performing this part of the experiment. Study. During the experiment, the rats were kept in a 12-hour light-dark cycle with food and water being available. During the brain stimulation reward experiment, surgery was performed in the rats to implement nonpolar brain stimulation electrodes. As for the condition-placed preference, rats were injected with either a vehicle, the control group, or the SB277011A drug along with cocaine or vehicle 30 minutes later. As for the reinstatement experiment, surgery was also conducted to implement intravenous catheters, which allowed the rats to self-administer an intravenous cocaine infusion. Injections of SB277011A was found to help block the enhancement of electrical brain stimulation that is produced by taking cocaine. SB277011A also reduced the effects of cocaine-induced condition-placed preference, as well as reducing the feeling of triggered reinstatement of cocaine addiction. This shows that by blocking the D3 receptor, it helped reduce both the rewarding cocaine effect and the relapse of cocaine-seeking behavior. The overall study demonstrates that by inhibiting the D3 receptor, this helps reduce the addictive properties of cocaine and suggests that further research of the blockage of D3 receptor can be a new prospect in treating cocaine addiction. After the use of cocaine, the amount of dopamine connecting to receptor in the nucleus accumbens can surpass the normal amount from natural activities, which produce pleasure that are greater than wanting food or sex. This addictive effect produced by cocaine is one of the many reasons that the relapse can occur. Other factors could include people wanting to escape from reality or feeling the need to try it due to peer pressure. Getting away from this feeling of pleasure can become difficult, and stealing life from the environment could pull people back into taking this drug. From the experiments, different drugs that inhibit different dopamine receptors, such as SB277011A and SCH39166, have shown to greatly, if not completely, stop the cocaine-seeking behavior. These results show that further research could potentially create a solution to the addictiveness of cocaine.